Okay, so in this lesson, I'm going to talk about sickle cell anemia versus unmutated hemoglobin. So in normal wild type, which is what we call any normal gene, a wild type gene, normal hemoglobin has the DNA sequence CTC, which you can see on the screen there with wild type hemoglobin DNA. When DNA is turned to RNA in the process called transcription, mRNA is complementary base pairing to the DNA from which it is reading. So the wild type DNA in this case is CTC. So the wild type mRNA is called GAG. In a second here, I will show you the mRNA codon wheel, which helps us determine which amino acids are going to be coded for by the mRNA. But GAG and mRNA, the codon, which is a sequence of 3 letters of mRNA, the GAG mRNA codon would code for glutamic acid, which is our normal amino acid in hemoglobin. The problem with sickle cell anemia is that there's a mutation in the DNA of people who inherit the sickle cell gene. So in the mutant hemoglobin, which you can see on the right, instead of having CTC in the DNA, there's actually CAC in the DNA. And this codes for mutant mRNA because the DNA code is what we considered mutant. So CAC DNA codes for GUG and mRNA. And using the codon wheel for mRNA, mRNA codon GUG codes for the amino acid valine, which turns the cell into sickle cell hemoglobin. So if you look at the picture on the left, normal hemoglobin has like a donut shape to it. It's got a concave center and the outsides are actually like spanned out. But sickle cell hemoglobin actually causes red blood cells to turn to like this sickle shape, which is like a half moon shape for the sickle cells. So how do we get to that? Well, the DNA coding strand you can see on the top there has a DNA template strand. And the DNA template strand is actually what is used for the transcription of the mRNA. mRNA is called messenger RNA because it takes the DNA message out of the nucleus and into the cytoplasm. So we use complementary base pairing to determine the mRNA sequence that codes for the DNA template, which we have in the nucleus of the cells. So in this case, our DNA template strand is actually the bottom strand, which you can see here is actually this bottom strand right here. And we use complementary base pairing rules to figure out what our mRNA strand is here. So in regular DNA, T binds with A, C binds with G, A binds with T, and so on and so forth. But when we're looking at DNA versus mRNA, we actually don't have T in our mRNA. And the reason for that is for the cell to be able to differentiate between DNA versus mRNA. So if mRNA has a U in it instead of a T, then the cell can determine that it's mRNA. So our DNA template strand has an A, which is going to bind with a U instead of T. G binds with C, T binds with A, C binds with G, and so on and so forth. Until you get back down to where you have an A again. In a regular DNA, the A would bind with a T, but we don't have T in mRNA again because T is for DNA only. The base uracil actually takes the place of T, thymine, in mRNA. So it helps the cell determine that when you have a U, it's actually mRNA instead of DNA, which has a T. So in this case, the DNA is going to bind with a U. So we have A bound with U, C bound with G, G bound with C, A bound with U, T bound with A bound with U, so on and so forth. So when we use the codon wheel, we actually use the mRNA for the codon wheel instead of the DNA. So the mRNA is going to actually be what we look at for the codons here for the codon wheel. So each sequence of 3 letters on the mRNA is called a codon, and the codon actually calls for the amino acids that's going to be brought to the protein. So in this case, we have UCA in our mRNA. So I come over here to the codon wheel. The first circle down here is where we start with the U. That's the first letter of the triplet code. So we start with U. Then we go to C, which is the second letter out in the second wheel. And then we go to A, which is the third letter out in the third wheel that you can see on the codon wheel here. And UCA codes for the amino acid serine. So to make sure that's right, we come over here to the translation portion, and we see that UCA codes for serine. So mRNA actually is used to translate the DNA code into a peptide chain. A peptide chain is a protein. So mRNA to protein is called the process of translation. And it makes sense because the language of mRNA is based in nucleotides, and the language of proteins is based on amino acids. So we go from nucleotide language to amino acid language in proteins. So UCA codes for serine. Let's just make sure that what we're talking about here makes sense. So GAG. I'm going to come over here to the codon wheel. I'm going to look for GAG, and I'm going to look for glutamic acid. Is the next amino acid in the peptide chain represented by glutamic acid? So I come over and look, and I see, oh, glutamic acid is the next amino acid in the peptide chain. Okay, so that makes sense. So now let's figure out what is the difference between normal hemoglobin versus sickle cell hemoglobin. So looking at what you've done so far in your lesson, you have turned normal hemoglobin DNA code into normal hemoglobin mRNA code. So as we said before, A is always going to bind with U, which is correct. T is going to bind with A, G is going to bind with C, G is going to bind with C, so on and so forth, making sure they're a complementary base pairing code until we get to right here, where you have C binding with G, which is correct, but then you have T binding with U, which if I look over here, T binding with A, T binding with A, T binding with A, so on and so forth. So this right here, this T bound with the U, is actually incorrect. This should be T bound with A on the mRNA because they're complementary base pairing. If you think about mRNA, T is actually equivalent to U in mRNA. So here we want to have T bound with A. So this should be GAG. So if we look back at the mRNA codon wheel, let's look back. So we want to look at CTC. We want to look at GAG. You have valine. Let's look at GAG. GAG actually codes for glutamic acid. So you have GUG here coding for valine. It actually should be GAG coding for glutamic acid. So normal beta hemoglobin amino acids should be TIR, HIS, VAL, ASP, SEER, GLY, and then you should have a glue here for glutamic acid. Then the rest of these are correct. Base pairing rules accordingly. Yep, Lu, Phe, and threonine. Okay, so coming over here, looking at the sickle cell beta hemoglobin DNA code, you're going to use the same rules that you applied in the last picture here for these pictures here. So A is going to bind with U in the mRNA code. So this should be this should be a U here. And then T should be binding with A, G binding with C, G binding with C, T binding with A, and so on and so forth. You should use the complementary base pairing rules to code your mRNA from your DNA. Then from there, you should use the codon wheel to figure out your amino acid sequence. So UAC is going to code for tyrosine, which you can use with the codon wheel. Now you'll notice that there was only one base pair change between the normal DNA code versus the sickle DNA code. And I want to have you find that out, but I can give you a hint. It's going to be somewhere over here. There's one nucleotide base pair that's incorrect over here in the mutant versus the normal code. So the fact that there's that one single nucleotide change, there is going to be a change in the shape of the protein. So that's what causes this normal red blood cell shape versus this hemoglobin, this mutated hemoglobin shape here with the sickle cell hemoglobin. So this last question here says, suppose the DNA had changed from CTC to CTT. So looking back here, what they're talking about is the CTT to the, CTC to the CTT. So this picture right here, what happened if this CTC would have changed to a CTT? Now you want to think about what would this DNA sequence have done to the mRNA sequence, and then what would that DNA to mRNA sequence have done to the amino acid sequence? You want to ask yourself, would it have changed it? Would it have made a big difference, or would it have stayed the same?